Hello, we are Team Airtech, and today we will be talking to you about our hypoxia gradient chamber. So to introduce you to our team, I'm Ali Heta, the team lead. This is William Lee, the facilitator, Cedric Milana, our software lead, and Munko Wong, the hardware lead. We also have our two mentors, Dr. Anya Grossberg and Dr. Wendy Liu. So to introduce you into what a hypoxia chamber is it's used to study instances of hypoxia in the body, such as myocardial infarction and cancer tumors, where a tissue culture is conducted under low oxygen environments. And so the issue with current hypoxia chambers is they don't really simulate in vivo conditions because they generate a low oxygen environment in one single concentration instead of a uniform gradient as seen in vivo conditions. And so now I'll be passing it off to our facilitator, William Lee, to talk about our approach.
Thank you, Ali. So as for our approach, what we will be using is a Clark electrode. It basically uses a platinum and silver chloride electrode in order to reduce the oxygen in the water into water. On the bottom right, you can see our design. The cavity in the middle is for holding the cells, and one side we will stick our electrodes in order to consume oxygen. Next slide, please. Now, the reason why we do this is that since we have oxygen that's at high concentration on one side through passive diffusion through the atmosphere, and since we have a Clark electrode on one side consuming oxygen, what this does is that this establishes a gradient throughout the cell culture chamber, and depending on where the cells sit in the chamber, they can exhibit different behaviors. Now, how we will validate this is through our mathematical model, both in MATLAB and in COMSOL, as well as cross-referencing it to what we see in real life. Now, I'll be passing this on to our software lead, Cedric Milano.
Thanks, Will. So for our product has to fulfill our acceptance criteria. It needs to be practical to set up and easy to use. It needs to be inexpensive to maintain and produce. It needs to be reusable, meaning that it can easily be sterilized and cleaned in between each use. And it needs to be able to be validated so that we can make sure that our gradient is always accurate to our specifications. Next slide. Our target market is any sort of tissue engineering lab that is interested in myocardial infarction or other similar hypoxia-causing diseases. Our target market is currently valued just under 60 million, expected to grow at a rate of 7.7% annually to a level of about 100.6 million. Our business model focuses on marketing and selling the hypoxia chamber itself first, and eventually, maybe later on down the line, selling culture slides and flasks that fit within that hypoxic chamber. And then after that, I'm passing it off to Munko Wong, our hardware lead.
Thank you, Cedric. So the verification and validation tests, we currently have 5 tests available. First, we set up the Clark electrode functionality test. Second, the Arduino system test, cell culture test, airtight seal test, and the platinum corrosion test. We were able to finish the airtight seal test, where we can see we submerged our device into dyed water and see if there are any leakage. Next slide, please. And here's our reference. Thank you.